In this video lesson, we're going to look at biochemistry and the environment. So first of all, we can look at sources of water pollution. So we could look at agricultural applications. So contaminating crops and soil could happen from the surface landfill leaching chemicals into the groundwater, which then gets into our households. The wind dispersion of particles from factories, runoff from the landfill can get into our surface water and contaminate fish, which then will make us sick, and so on. So there's lots of different ways that pollutants can get into our water system. So xenobiotics kind of sounds like antibiotics, but it's not. So these are chemicals that are found in organisms that aren't normally found there. So they can come from landfills or improperly disposed waste. Some examples of xenobiotics that we are exposed to would be plastics, microplastics predominantly, pesticides, medications that are improperly disposed, solvents that have been poured down the drain and end up in our water system, and so on. Some of these xenobiotics can be metabolized by the animal, so if we can break them down, they're not going to continue to cause harm. But others will remain forever, often because they are nonpolar and then get absorbed by our fatty tissues, and they can actually affect our metabolic processes. So the first type of xenobiotic that we'll explore is actually antibiotic waste. So these are improperly discarded antibiotics, and they can leach into our soil or water, so homes and hospitals could disperse them, and then they end up in our water. Could even be from a pharmaceutical company dumping their chemical waste. Farms are a big one, actually, because farmers will give their livestock antibiotics to keep them healthy, prevent them from getting sick, and then their waste will end up being released. And we even could use their waste, their manure, for fertilizer for crops, so then we're exposed that way. And then it gets drawn in by a drinking water treatment plant, and then we eat it. So it's a problem. However, water treatment plants can remove antibiotics in a few different ways. So they could have activated sludge, and that will have bacteria that can actually break down some antibiotics. This one isn't as effective as carbon filters or ozone. But carbon filters and ozone are very expensive. So depending on the town or the city, you may or may not have the more effective method for removing the antibiotics. Way back in grade 9, when you studied ecology and the sustainability of ecosystems, you learned about biomagnification. So that's when you have something, a xenobiotic, amplifying itself through the food chain. So some materials don't break down and they remain in the tissues of an organism. So when that organism is eaten by another, the accumulated chemical magnifies. So you can see that happening in this diagram. So that the top carnivores, at the end of the food chain, they're going to be most affected because they're eating the thing that has a lot accumulated within, and they're eating many of those. So it continues to magnify through the food chain. It accumulates, so bioaccumulation is when it's accumulating in the tissues of the animal or plant, and then it magnifies through the food chain. So nonpolar molecules are things that do biomagnify because they are accumulated in the fatty tissues of the animal or organism. Pesticides are an example of something that can biomagnify. So one pesticide that you should be familiar with is DDT. Here's the structure. It's a pesticide that was widely used because it was effective. It did the job it was supposed to do. Plus, it was colorless, tasteless, and odorless. So if you applied it to your food crops, nobody smelled it or could taste it or see it. So in some ways, it was considered a good thing. But because it was so widely used, insects developed a resistance to it, so you had to use more and more of it. And it was actually used for about 100 years before scientists learned that it was linked to biomagnification. And in 2004, it was banned for all use except to eradicate disease-causing mosquitoes. So in some places in the world, it is being used still to kill off mosquitoes that cause malaria. So it's the lesser of the evils. Malaria kills so many people every year, so to eradicate the mosquitoes would be a good thing. Another thing that can cause bioaccumulation are heavy metals. So metals can accumulate in your tissues, and because they're elements, they're not broken down, so they remain in the body and they don't get excreted. So one example of a heavy metal is mercury, and that gets converted to methylmercury by aquatic organisms, and then fish eat it and they build it up in their tissues. And methylmercury is nonpolar and insoluble in water, and the really big problem is it's because it's so small and nonpolar, it can pass through the blood-brain barrier and across the placenta and cause neural toxicity. And one example in Canada where mercury poisoning was a huge problem was in Grassy Narrows, which is a community near a pulp and paper mill. And the people that lived in the community suffered tremendously by the industrial waste that was poured into the water system, and the people developed mercury poisoning, and it took many decades for the Canadian government to acknowledge that there had been this happening and that there was a cover-up, and that the people did indeed suffer from mercury poisoning at the hands of the government. Another xenobiotic is plastic. So plastic is cheap, it's convenient, and it's a durable material. And it is necessary in many instances. It can be sterilized and used in medical settings and then be disposed of to prevent cross-contamination and the spread of disease between patients. And it can be made very easily from addition reactions to create polymers. And many plastics can be recycled, but large quantities, even if they are recyclable, end up in landfills and unfortunately into the ocean. So some of the plastic that end up in the oceans end up in these areas called gyres. So the ocean currents will carry them, and then they basically are in these gyres. There's actually 5 of them. There's the South Pacific, the South Atlantic, the Indian Ocean Gyre, the North Atlantic Gyre, and the North Pacific Gyre. And what happens is if the plastic just stayed in its unaltered state, that would be not great, but it wouldn't be as bad as what really happens. So sunlight degrades the plastic, but it only breaks the plastics into shorter lengths of the polymer. It doesn't actually decompose it or get rid of it. So now you have tiny pieces of plastic, some of them imperceptible, floating in these gyres, and then wildlife will eat it, and that's a problem. However, there is a bacteria, Ideonella zacchiensis, is a bacteria that can actually digest PET plastic, polyethylene terephthalate, and it releases an enzyme called PETase, which breaks the plastic into its monomers, which then the bacteria can digest. So this is a good thing. So if we can find a use for these bacteria to break down plastics, that would be wonderful. Another solution is to use bioplastic. So plastics are made from starch, cellulose, or biopolymers rather than from fossil fuels. And then these plastics, because they're made from starch, can be digested by regular bacteria in landfills. So you won't need this specialty bacteria. There's another technology known as bioremediation, and that's using the process of using bacteria to digest harmful or toxic compounds into smaller, less harmful ones. And it uses something known as the host-guest interaction to work. So the host is usually an enzyme, the guest is the toxin. It's kind of like our lock and key model for enzyme, the enzyme model. The two will interact by intermolecular forces, so it could be London forces, dipole, however. And then the host will metabolize the gas, so break it down into less harmful compounds, and then we've remediated the soil or whatever we're trying to remediate. So one example would be host molecules can be synthesized to remove a specific guest molecule. So one example would be removing radioactive cesium-137 isotopes from nuclear waste. This is the molecule that can do that. You don't need to know the structure or name, but just that you can use them to remove specific guest molecules. Scientists have also found ways to clean up oil spills, so specific enzymes have been synthesized to clean up oil spills. They surround the oil droplets, break them down into carbon dioxide and water, and then the oil spill is remediated. So there is a table from our textbook in the Biochemistry and the Environment section that shows a few different types of enzymes used to clean up industrial waste. So proteases obviously work on proteins, and their job, they could be used to clean up excess food waste, leather and pharmaceutical waste that's been disposed. Cellulase works on cellulose, so that can work on textiles and pulp and paper. And then lipase works on lipids or fats and oils, so we can use that to clean up, food waste, cosmetic problems, pulp and paper as well. Detergents have now been used to include enzymes that break down food particle or other waste or dirt particles into smaller molecules. These detergents, because they have these enzymes, can now allow the clothes to be washed at cooler temperatures and using less detergent and less pre-treatment of the stain. So you can actually get your clothes cleaner in colder and less water and less work. So that's a benefit. So there's a concept known as green chemistry, and the concept of green chemistry is to use less energy and generate less waste. So some of those examples are shown in our Cogni option, section 6, table 1. So one green chemistry principle is to just prevent waste in the first place. So if you generate less waste, you don't have to clean up the waste. So that's good. The second one, which we'll look a little bit further at, is atom economy. So in the synthesis of a compound, you want all the materials used in the production to be incorporated into the final product, rather than having leftover materials or byproducts as waste. Using less hazardous chemical design and synthesis makes sense. If you're using compounds with little or no toxicity, we should use those rather than things that do have toxicity. Have safer and restricted use of solvents. So safer solvents should be used when you can, and you could recycle those solvents or reuse them, and then in fact, using no solvents at all would be the preferred method. And then efficiency of energy. So using less energy to make the thing would be helpful. So some examples of green chemistry. So using enzymes to have targeted results, because enzymes are specific to substrates. Choosing the right solvent. So allowing the reaction to take place in the right medium, whether it's a non-polar solvent or a polar solvent. Doing things on small scale produces less waste. Being energy efficient reduces the waste of energy. Having safer waste, rather than washing harmful waste down the drain, we can autoclave things to sterilize things, so then we're not producing the same amount of waste. So an autoclave is almost like a heat sterilization instead. So atom economy, the goal is to use up all the materials in the production of the product with no waste, as I mentioned before. It factors in, though, the mass of the catalyst, but not the solvent the reaction happens in. If the atom economy is near 100, then it's classified as a green process. And the way you calculate it is you take the total mass of the desired product over the total mass of reactants times 100. This is an example found in our Cognitive textbook. So in this example, we're making malic acid from benzene. And again, this is the reaction, and they're using mole ratios and molecular masses to calculate the atom economy. So the total mass of the desired product would be 192 grams, because they want 2 moles of malic acid, times the 96 grams per mole, so you end up with 192 grams. The total mass of the reactants are 2 moles of benzene times 78 grams per mole, plus 9 moles of oxygen times 32 grams per mole, so there's 440 grams of reactants, producing just 192 grams of the desired product. The rest is water and carbon dioxide, which aren't hazardous compounds. So in this example, the atom economy then is the 192 over 444 times 100, only a 46% atom economy. So there are limitations to using this as a measure, because in the example we just looked at, carbon dioxide and water aren't target products, so they're not included in the calculations, and that made the yield really low, saying that it wasn't a green process. But again, the products aren't harmful. And then also in atom economy, catalysts are included, but they get reused, plus they provide a more efficient pathway than an uncatalyzed reaction. So again, this atom economy model doesn't always work. It doesn't work because we are looking at things that are reused, the catalysts, and some of the products aren't harmful. So sometimes it'll make something look like it's not efficient, even though it is.